Hello, everyone. Thank you for attending the first episode of Healthcare and Research Ethics for COVID-19, Possible Treatments and Vaccines. I'm Kelly McBride Folkers, the project manager and member of the NYU Grossman School of Medicine's Working Group on Compassionate Use and Preapproval Access, also known as CUPA, and I'll be moderating today's event. Our mission is to advance research, policy, and education regarding the ethical issues surrounding access to investigational medical products. We're proud to present the first episode of our webinar series with the University of Arizona James E. Rogers College of Law Health, Law, and Policy program. Before we begin, I'd like to share some housekeeping notes. All participants will be muted for the duration of the webinar. We will have 5 minutes for questions after each segment of today's event, and we ask that you please enter your questions for our panelists in the Q&A function. You should find an icon below the video panel on your screen in Zoom that says Q&A. Please click on this icon and you will be able to answer your questions. We will have a moderated Q&A with all panelists at 2:45 p.m. Eastern Time. Now I would like to introduce our first speaker, Dr. Arthur Kaplan, who will be speaking about the ethical issues in the development of a COVID-19 vaccine. He is the Drs. William F. and Virginia Connolly Mitter Professor and founding head of the Division of Medical Ethics at the NYU Grossman School of Medicine. Along with Dr. Allison Bateman House, speaking later today, he serves as the co-chair of CUPA. Take it away, Art.
Well, thank you, Kelly, and thank you, everyone, for being here for this webinar. We have a very, very interesting and timely topic looking at the development research on vaccines to try and battle the COVID-19 virus. There are huge ethical questions involved in this effort, and we'll review some of those and try and make clear what some of the options and alternatives and ways of thinking through the challenge of getting to a vaccine involves. So I'm going to review with you a little bit of how vaccine development historically has worked. I'm going to say a few words about why that has been very slow. I'm going to then tell you some of the obstacles that we face in doing research to develop a vaccine. Then I'll suggest one possible strategy for speeding up the development process, which is a so-called challenge study, and give you some of the pros and cons of that. And then we can finish with some of the issues around trust and public acceptance of a vaccine, which is a major issue as this particular vaccine research goes on. So go all the way back. Many of you will have heard pronouncements from the president, from scientists, from people working on vaccines, from corporate people saying, we hope that maybe we could get a vaccine by the fall. Anthony Fauci and other infectious disease people talk more about 12 months to 18 months. I personally think these are very optimistic projections. I cannot imagine us having a vaccine available to give to the world by the fall. I worry that when that particular time frame is announced, it gives the public false hope. They may wind up not trusting claims about vaccines as much if people keep saying, we hope to have something ready to go by the fall. And obviously saying you have a vaccine by the fall gives people hope that the bitter days of quarantine and the damage it has done to their pocketbooks through economic damage will finally be over, because we'll be able to vaccinate our way out of this particular pandemic. I think a more realistic time frame comes in years, not in months. And here's why. In developing vaccines, you have to follow a process that's somewhat similar to that used in developing drugs. You find yourself first trying things on animals, usually primates, for vaccines to see if they develop antibodies and to see if there's any adverse events. Basically, the idea of a vaccine is to create immunity, not by giving you the disease per se, but by giving you a diminished version, sometimes called attenuated version of the virus, a weakened version, so that it doesn't make you sick, but it hopefully triggers an immune response in the vaccine recipient. or by giving you bits of DNA that might be involved in the virus's code that could trigger, or RNA for that matter, either way, genetic information that might trigger a response, again, of the immune system without any risk of giving you the disease. So hopefully we will find that people make immunity to the COVID virus. On the other hand, we don't know that yet. So one of the biggest obstacles is, will we develop sustained immunity if we get infected by the COVID virus? Will we develop immunity sufficient to battle it if we get a vaccine? And hopefully we will, but again, there's not any guarantee that you're going to be able to build sustained, durable immunity, either through natural infection. You know, we all get colds and they come back and they infect us again and they infect us again, and that's a historic problem with this family of virus. Or we get an injection and we build an immune response, but it's not big enough or strong enough to take on and kill the virus that gets into our body. That's been the problem with, say, HIV. We haven't found a vaccine capable, despite looking for many, many decades, of generating up an immune response sufficient to knock out all the different strains of the HIV virus. So, first big challenge is, do we find something that actually works? The second big challenge is it safe? We are talking here about a intervention, a medical intervention that would be given to hundreds of millions of people in the US and billions of people worldwide. When you're developing drugs, you certainly are attentive to adverse events and things that the drug might cause in terms of unwanted side effects. But for almost every drug, you don't plan to give it to the entire population of Earth. So you know that if you're going to give it to someone, say, with cancer, or someone with ALS, or someone who has depression, you're talking about a narrow subset of the human population. It still may be a big group, but it's not every child, every elderly person, everybody with comorbidities. Vaccines go out to huge swaths of humanity. And this one would probably be the biggest footprint of all in terms of trying to give it to everyone to get rid of the disease, probably the biggest footprint since polio, where you're trying to vaccinate the world against that particular plague. So you have safety issues that really have to be carefully managed, because even though you might accept a risk of, say, 1 in 50,000 having an adverse event, if you're giving it to 3 billion people, you're talking about a lot of adverse events. So you have to test vaccines at bigger numbers, larger numbers of people, so that you can be sure that you really have something that both is generating an immune response, but is also safe. In drug trials, once you move from animals, you do the same thing with vaccines. You probably do basic safety studies in maybe 20 people just to make sure that everything is okay. Then you move on to phase 2, as the FDA calls it, in the US regulatory system. We're starting perhaps to settle on a dose to see how far you could go without causing an overreaction of the immune system, which has its own problems. And then you come to phase 3. In drugs, you might be testing 1,000, 2,000 people who have a particular disease, and you're going to see whether the drug helps or alleviates symptoms or does something for that particular group. With vaccines, you're probably talking 20, 30, 40,000 people minimum to see what happens in large populations, so that you can detect both whether everybody builds the same immune response, or some people don't due to genetic reasons or age or underlying disease, or because they're children, and whether in fact you've got something that really is very, very, very safe. Nothing is going to be 100% safe. Even standard vaccines long used that have been recommended, they may have 1 in a million kinds of dangers that go with them. But for this vaccine, I think you're really going to have to get strong evidence that it really is safe. So that takes a long time. Why? Because when you move to phase 3 studies, you have to answer a number of questions. One is, will a single shot build up enough immunity, or are you going to have to administer more than one shot? Think of the HPV vaccine that is used to treat prevent cervical cancer or prevent genital warts, that is a 2 or 3 shot vaccine. Cholera is a 2-shot vaccine. Hep B is a 2-shot vaccine. It may be that giving somebody a single shot of something that builds immunity to the COVID virus requires something to follow up to boost up the immunity even more. If that's true, that's going to take a long time to figure out, and it's going to take a long time to manufacture, let's say in the US, not 300 million vaccine doses, but 600 million vaccine doses or more. So huge challenge in terms of speed to get that kind of answer. You also want to know how long does the immune response last? How durable, as I said earlier, is the response? Flu, we seem to have to vaccinate every year, partly in honesty because the different strains of the flu come around, but partly because it doesn't look like the immunity lasts. So that becomes an annual task. Other vaccines have, boosters that have to be administered for them. So again, you're looking at bigger challenges in terms of manufacturing and distributing with gigantic numbers, billions if we're talking about the world, potentially having to go back annually and do it again and again and again. That would be a staggering logistical nightmare in terms of availability of a vaccine. Other challenges become effectiveness. Vaccines are great, and they, in my view, have been the biggest public health invention success in the history of humankind, maybe sanitation and clean water, but right after that come vaccines, prevented more deaths, more disability than any other intervention, anything that medicine and science has ever done for humanity. But no vaccine is 100% effective. For biological reasons, some people don't respond to vaccines when most people do. Some people, the elderly often have weaker immune responses as they age. Their immune system isn't as effective. Newborns don't have much of an immune system going when they first get into the earth, when they first appear. So again, you have to wait to give them injections frequently at 6 months or a year so that their own immune systems are there. They're relying on the mothers passing on immunity to battle microbes and germs. In any event, probably one of the most effective vaccines we have is measles. I believe that's 94 or 95% effective. Whooping cough might be 85% effective. The flu vaccine in a good year is probably 50 to 60% effective. In a bad year, maybe 30%. So if we find something that seems to work, hopefully it will be much more like the measles vaccine, but it may not be. And if it isn't, then we have a problem because we're not going to really get herd immunity out of something that is only, let's say, 50% effective. We'll also face a policy choice. If we find a vaccine that is 50% effective, are we going to make it and distribute it worldwide? Is it good enough? And how that question gets answered and settled, there's going to be a lot of debate among experts, among governments, among all manner of people. But if you roll out a 50% effective vaccine, you're devoting the available capacity of manufacturing to make that, and it's hard then to say 2 years later, oh, we got a better vaccine, it's now 80%. So for all of these reasons, I am very, very, very skeptical that anything will be available in any kind of numbers in 6 months, 12 months, or 18 months. You have both the research path to pursue to demonstrate safety, efficacy, durability, how many shots you need, how effective this thing is, do you need a booster? And then you've got distribution issues. You got to make it in huge amounts and distribute it both all over the US and all over the world. These are just incredible challenges. I can also tell you, having studied vaccine development for many years, it's pretty rare that something doesn't go wrong in the manufacturing side, either in the clinical trial process or in the manufacturing process. A plant goes offline due to glitches and whatever, and you have to shut down and diagnose the problem and get the biological incubator or the assembly line, depending on what you're making, back up and running. And those are notoriously accident-prone manufacturing processes, and they frequently do break down, adding time, again, to the availability of vaccines as you try to make sure your manufacturing capacity stays intact, both for supplying it to research subjects, the 30, 40,000 people that you're going to study, or to distribute. Well, perhaps one of the core reasons why it takes so long to get an answer on safety and efficacy in clinical trials with any vaccine is that you give people, which takes time to ask them to be in a study of the vaccine, and then you are waiting after you get them all injected for nature to infect them. I mean, by that is you're relying on natural attack by the virus just out in the wild type world to infect the people who've given the experimental vaccine to. That can happen quickly depending on how prevalent the virus is, but it can take a while if hopefully rates of infectivity go down, if we bend the curve downward, if for whatever reason the virus decides to ebb and flow and not be present all the time, then that lengthens out how long it takes for people to get infected. So you're waiting and waiting for infections to occur and then seeing whether the vaccine helped or did it have adverse consequences once the virus got into the body and the body's immune system begins to respond. The normal time for that kind of clinical trial is measured in years. I don't really see much way to speed it up. There are some vaccine manufacturers right now who are nervous that they're going to run out of subjects in the UK or the USA as as viral infection rates begin to drop down. And remember, another ethical duty with your subjects is once you immunize them with the experimental vaccine, you do have to tell them how to avoid getting infected. You really don't want to say to them, oh, well, we're not going to remind you about washing your hands or staying socially distant or wearing a mask or other personal hygiene measures that they can take to avoid becoming infected. It's morally our duty to protect them from infection, since we don't know if the vaccine's going to help them should they get infected. So we can't just say, do what you want, expose yourself, try to avoid taking care of yourself. That wouldn't be the right thing to do, and it isn't done in these clinical trials. So one idea that comes up for speeding up this aspect of getting answers about safety and efficacy and providing good data is to deliberately infect a vaccinated person with some version purified of the COVID virus. In other words, deliberately infect a healthy person who has been given the experimental vaccine with an agent that we know causes death and hospitalization and perhaps can cause longer-term disability that we don't even understand. So this is highly ethically controversial. The upside is the world is ravaged by this plague, economy's broken, people quarantined, they can't really take the isolation and the being locked up. The behavioral ways of coping with this outbreak are very difficult to live with, very difficult to enforce. So we have a likely ongoing plague with many people becoming sick and dying, and economies basically in trouble, as a nervous public doesn't even want to come out. Many won't go back to work. Many won't support industries like the theater or mass events like sporting events or concerts. Damage to the economy is occurring across the board as people don't have confidence that they can safely return to normal. If you deliberately infect people, you can take a smaller group than 20 or 30,000, you probably could do this in the hundreds, put them in a place where they can be observed and watched closely as they get exposed to the challenge version of the virus. Make sure there's hospitals handy, make sure they have every possible therapeutic agent that might look helpful should they become sick. There hopefully will be more of these kinds of therapeutic agents that might do a bit of good by the time this kind of challenge study got underway. And you want to make sure that those people have those available. You also are going to try and recruit, I think, to minimize the chance of people really getting sick if they do get infected and the vaccine doesn't work. That means probably younger people, we certainly want an answer to whether the vaccine would work in older people. You'll look at the damage done in nursing homes to people in terms of deaths. It's just been staggering, and obviously vaccination there would be highly desirable, but that's not a group you're likely to start out with either in a clinical trial or in a challenge trial. It would be harder to tell what's going on in terms of adverse events, and I think the risks might be unacceptable in terms of exposing much older people to vaccines, and then in a challenge study to the actual virus. You might recruit among medical students, you might recruit among the military. Interestingly enough, the numbers get smaller, and if you do the challenge study as opposed to the longer traditional clinical trial, you don't have to worry as much about, are we going to do a placebo group with sham injections or not, or can we just use as a baseline an observational group in the clinical trial? And if the first vaccine doesn't work, you can shift over very quickly to the next promising vaccine. Remember, launching a clinical trial for the first agent that looks good, many of these are likely to fail. So you're talking about sequentially trying to recruit tens of thousands of people, wait for nature to infect them, and moving from one vaccine to the next. Again, that's why I say if you're going to do it that way, 6 months, 12 months, even 18 months, it just doesn't sound reasonable in terms of trying to get what we all hope for, which is a successful vaccine. So even if you go at Operation Warp Speed, the president's program, or like Britain devote a lot of money to vaccine research, China doing the same, other countries as well, you still, if you follow the traditional path, you're going pretty slowly and you're moving from one hopeful agent to the next without any promise that the first one's going to work or work with a big enough response that you don't have to use boosters for it and so on. So even if 100 or more groups are looking, you still have a long testing tail here. So I do personally favor trying the challenge studies with conditions. One is you absolutely have to have the informed consent of the volunteers. They have to be motivated by altruism. I probably wouldn't pay them, so there's no confusion about why they're doing it. You'd have to make sure they comprehend all the risks and the unknowns fully. I'd probably give them quizzes to make sure that they really understood. I'd give them a chance to change their minds, probably consent them twice before I put them into the study to make sure they really are ready to take on the dangers that they would face in being in a challenge study. And I think we want to make sure that while we get some ethnic and racial diversity in the study population, I would not be using people who seemed overly eager to get into this, maybe ignoring the risks or blind to it. It's great to want to volunteer and help, but maybe we have to be a little cautious about the competency of people and checking them either psychologically or psychiatrically. But if the informed consent can be done and done carefully and done well, then I think letting people take these risks is some letting people donate a kidney or a lobe of liver or work in dangerous occupations. I even think the risk of this is less than saying to a healthcare worker, keep working around infected people, even though there's a pretty good chance if you're in a nursing home or in an ICU or intubating people that you are going to get infected. So comparable risks seem to me acceptable relative to what we permit other people to do in other settings. We have done challenge studies before. It's not a long list of challenge studies, but they have been done. Even when rescue therapies have been uncertain, say malaria, that we could really pull you back if you got it for the malaria vaccine. But in any event, I think the risk benefit in the midst of a plague with the tremendous death and damage that it's doing justifies seeing whether people would volunteer. So far on websites, about 25,000 people have said that they would do it and counting. They're all over the world. They're not in one place. We'd have to bring the group of people that we were going to involve in this to one place, observe them, give them the best available access to medical care and so on. But nonetheless, there's at least some reason to think that there are enough folks out there who would be willing to volunteer to help, that this challenge study model could be done. If we're going to do it, we'd have to begin preparing the pure vaccine now, because that would take a couple of months to get ready and test it for safety. So those who favor that option, like I do, want to begin not so much tomorrow morning with the challenge study, but begin the preparations in terms of where the people would stay, getting a virus, an artificial virus ready to go to test against the vaccine agents and so on. The last part of the story about developing a vaccine is once you get it, whether by clinical trials or using challenge trials, if something really does look safe and effective, then you have public trust. If the public thinks that the vaccine was rushed or the data is weak or uncertain or less than overwhelming in terms of safety and making sure that no one's going to get sick from it, there are a large number of people, both in America and elsewhere, who may not take the vaccine. There's already plenty of discussion on the internet about fighting mandates against vaccines, about conspiracy theories about why vaccines should not be taken, whether Bill Gates has made them so that he can profit, or that cell towers are involved in transmitting the virus, or that vaccines are somehow a control agent of governments, and on it goes. The anti-vax movement was getting stronger before this outbreak in terms of getting into fights about measles vaccination, childhood vaccinations, and so on. They've already started to transfer their opposition to vaccination to the potential, not even existence yet, COVID vaccination. And there are many other people who would just look at a vaccine and say, I don't know, they seem to have rushed that. Didn't they talk about this as somehow fast-tracked or warp speed or done very, very quickly? That makes me nervous. I'm not sure I'm going to take that. So again, another obstacle to vaccine research and vaccine development and acceptance is public trust. If scientists were to say, well, the evidence isn't as good as usual, but we're in tough straits and we're going to roll it out, I'm not sure that's going to gain the kind of public support that you would get if you had strong data, convincing data. Challenge study is going to give you strong data and convincing data quicker. If you're doing clinical trials and you're on your fourth vaccine that hasn't worked and you're starting to really panic, maybe that could get you in trouble. I will say historically, both the polio vaccine and swine flu vaccine, when they were rolled out, did have manufacturing issues because speed was of the essence, and public distrust, including the episode under Gerald Ford with the swine flu vaccine manufacturing problems, led to some of the momentum behind the anti-vaccine movement. So that has to be watched very, very, very carefully. Finally, again, even if you get this vaccine and the data is good and it looks strong and you don't have to give it every year and it gives you sustained immunity, and but if we got it, you still have to manufacture it and make hundreds of millions of doses in the US, billions of doses around the world. It's not going to come out at the same time. People will have access to the vaccine as it gets made. It'll roll out. That will raise questions about who gets it first. There may be many appeals about compassionate use or groups that feel they have an emergency need, healthcare workers, first responders. That'll take us into topics later in the webinar about how one makes decisions about which people or which individuals might get first access to a vaccine as manufacturing proceeds. And then distribution, we have to watch out for justice. Many nations are going to say, we're making vaccine and we're keeping it. And we don't care whether the outbreak isn't here, we're going to use it here first, even if it would make more sense to use it in a country that was having an active outbreak and you were trying to control spread from there. There is no real international authority or kind of power that can settle how countries decide to divide up the supply of new vaccines, either in terms of who gets it first or what countries are going to be able to access first. Remember, many of the manufacturing plants are not in countries that are funding the research. Some vaccines are made in third world countries. Some vaccines are made, say, that the US might be sponsoring, but they may be manufactured in Belgium or in Britain or France or some other place. And those governments may say, we're not sending vaccine to the US until we vaccinate our populations first. So that's a justice issue around distribution that we have to be alert to as well. So in sum, people talk about a vaccine being developed in 6 months, 12 months, 18 months. I think we would be lucky if we got a clinical trial the first time out of the box that showed something safe and effective after 2 years, 3 years. We can't chintz on the demonstration of safety and efficacy because you'll undermine public trust and you'll open a window for anti-vaccination sentiments to undercut willingness to use this vaccine, which is our solution to this particular plague. Challenge studies might speed up the research process, but they're ethically hyper controversial and should be, because giving people dangerous agents violates do no harm standard principles, but perhaps in a worldwide plague with the damage being done, it makes sense to bend that principle in order to get an answer more quickly about whether there is a safe and effective vaccine out there. And even if we find something, when somebody says 6 months, 12 months, I think they're talking about something that looks promising, not manufacturing it by the hundreds of millions of doses, much less the billions of doses, much less something that requires 2 shots or 3 shots. So that's why I say I don't think the world will see a safe and effective vaccine against the COVID virus for years, not months. So I want to thank you for watching my presentation. Hopefully we'll get some questions and comments based on this talk, and I look forward to seeing those.
Okay, thank you so much, Art, for a very comprehensive overview of the state of COVID-19 vaccine development. We have a couple of questions. As a reminder, please enter them in the Q&A function at the bottom of your video panel. We only have time for one, so I'm gonna take this one. Are there previous examples of using challenge studies in vaccines, or would this, the development of COVID-19 vaccine using a challenge study, be unprecedented?
Well, Kelly, there are previous examples. Typhoid, cholera, and flu have all been done with challenge studies. And it is interesting, no one died in any of those studies. There are also challenge studies of a different sort, which we forget about, but are not so uncommon. And that is testing environmental pollutants on people to see what happens, or even household products like bleach or deodorant or perfumes. People get tested for that too. And there have been deaths in those studies, but the only way to check the safety of something is to give it at some point past animals to humans and see how they respond. You're trying to get a mild response that indicates any danger, but sometimes it gets out of hand. So challenge studies, not common, I don't want to suggest that, but yes, they do have a history. By the way, the first one I should mention was Edward Jenner and his smallpox vaccine. I wouldn't say this was ethical, but he grabbed the kid of the gardener, gave the kid his inoculation, and then deliberately infected the poor 9-year-old with the disease. So that goes back to 1880. Not a great precedent in terms of exploiting the poor, but there it was.
Great, thank you so much, Art. So I see there are more questions coming in. I'm going to note these for Art, but we do need to move on. All right, our next topic is access to unproven therapies for COVID-19 patients, clinical trials, emergency use authorization, and expanded access and off-label use. And we're going to hear from some more CUPA members. I'm pleased to introduce my colleagues. Professor Christopher Robertson is the Associate Dean for Research and Innovation and the founder of the Regulatory Science Program at the University of Arizona James E. Rogers College of Law, and we'll hear from him first. Next, we'll hear from Dr. Allison Bateman-House, the co-chair of CUPA, and she is an assistant professor in the Division of Medical Ethics at the NYU Grossman School of Medicine. Dr. Bateman-House will be teaching a course at the University of Arizona James E. Rogers College of Law on access to investigational medical products, clinical trials, expanded access, and right to try this summer. Finally, we'll hear from Professor Holly Fernandez-Lynch, and she is the John Russell Dixon, MD, Presidential Assistant Professor of Medical Ethics and Health Policy at the University of Pennsylvania. Chris, you have the floor.
Thank you so much, Kelly. It's great to collaborate on this project from various places all over the country, and I think maybe all over the world. So it's great to be here. I do have a few slides to share as I speak for just a few minutes. So I hope you can now see my screen. Someone will call out if not. So I'll be talking about intended and off-label uses of drugs, especially in this COVID context. And for that discussion, my presentation will be these first two bullets about how a new indication is approved by the FDA, and then how other indications, or other uses as we call them, can nonetheless be used by physicians in this way we call off-label. In the next two talks by Allison and Holly, will go into two other pathways essentially to get access to new indications or unlabeled indications. Pre-approval access, which includes both expanded access and right to try, will be Allison's topic, and then Holly will be talking about emergency use authorizations. So interestingly, in the COVID era, these are all sort of intertwined in really interesting ways. But this really starts with the rather unique feature of the Food, Drug, and Cosmetics Act, which really creates a pre-market approval system. I mean, think about it. When you go to buy a t-shirt, or when you go to buy a television or a car, no government agency has first reviewed that product to determine whether it can be marketed. Rather, instead, we use post-approval systems of regulation if there's a complaint, or even liability. You can go to court if someone's injured after the fact. But what's kind of special about the FDCA, the Food, Drug, and Cosmetics Act, is that it has this pre-market approval system where you first have to apply to the Food and Drug Administration. Now, what's interesting is the threshold requirement for what requires for when you have a product that requires that approval is really just anything that for which you intend it to be used in diagnosing, curing, mitigating, or treating or preventing a disease. And you can imagine these are legal terms, so all of those have been litigated, and there's some others too about affecting bodily function. But here's the core concept I want to emphasize, is that the very notion of a drug, you know, a product like a can of turpentine, for example, becomes a drug only if the seller or the manufacturer or the shipper of that product intend it to be used in this particular way. Otherwise, it's just a chemical compound or another sort of product. And so once you have that intent and it triggers this need to file an application, the Secretary of the Health and Human Services, or his designee in the FDA in practice, has to decide whether to approve that application. And here again, the law gives two primary considerations. First, whether it's safe, and second, of whether there's substantial evidence that it will have the effect it purports to have on the label. So this are together called safety and efficacy. If not, you have to approve you have to refuse the application. And of course, to prove safety and efficacy, typically a drug goes through three phases of clinical testing, and by clinical testing we mean testing in humans, starting with a handful of participants and moving forward to hundreds or even 1,000 or more participants, depending on the study. Then the FDA reviews that data to determine whether it has found safety and efficacy. And if so, it sometimes requires what's called those phase 4 post-marketing studies, which are especially important for finding rare adverse events that may not show up if you only have 1,000 people in the phase 3 trial. But that's that process of proving safety and efficacy. And I just want to emphasize that it's linked to the labeled indications. And it's really important that we have a sort of alignment between the labeled indications, what the drug is sold purporting to do, and really what the drug maker intends. We don't want a drug to come on the market where the drug maker intends it to cure cancer, but it's really only been labeled or tested for its ability to reduce high cholesterol. That alignment is really important for the regulatory system. And so these are three concepts I've now introduced to you already, and I just want to show how the company's intended uses, the ones that have been proven safe and effective, and the labeled uses, the function of the law is to make sure these come into alignment so that the labeled uses are all the ones that have been intended by the manufacturer and proven safe and effective, and only those. Now, so the reason for this is, of course, historically, it's been a concern about what can be pejoratively called paternalism, to protect people from dangerous products or drugs that are cost money and don't have a benefit. But I suggest there's this larger function that we always have to remember, which is the production of knowledge. By telling companies to prove it before you promote it, it gives the companies an incentive to invest in that scientific testing, and so that they can prove whether it does have any value that they intend to exploit. And this is the way we align our financial expenditure and our commitment of our bodies, that when we give a drug into our bodies, that's how we assure that it will have some value for that purpose. Now, that background was necessary to explain this concept of off-label use, because that notion of alignment shows that drug companies may only promote uses shown on the label. And the reason for that is if you were to promote another use, it would reveal that the drug has other intended uses that haven't yet been shown safe and effective, and then which aren't shown on the label. And so the company's marketing and promotion, here defined broadly to include everything from advertising to detailing, really has to reflect that same alignment. I have an asterisk here to recognize that there's actually some litigation and theory about whether the First Amendment agrees with this theory I'm now promoting. I'll show you some articles you can read about that for further interest. But the next step for our purposes today is that doctors can prescribe drugs for other uses according to their own discretion in state law. When the FDA was created, there was this notion of federalism, that the practice of medicine was really a concern for states, and that we didn't want the federal government interfering in that practice. And so this was in part written into the federal statute, but remains a sort of background commitment of the FDA that doctors are free to use their discretion. And sometimes that's valuable because doctors will have a sense of expertise that's not yet reflected in the scientific basis or in the company's testing. And so this has forced them, this limitation on companies from promoting off-label uses really forces companies to instead prove additional uses safe and effective through additional study. And so, in fact, about half of what the FDA does in any given year is actually review applications for expanding labels as companies produce that additional scientific evidence that the drug is safe and effective for other things. They may have initially gotten it approved for colon cancer, but they get a supplemental indication for, say, breast cancer. And that's all to the good if it's produced additional knowledge that then physicians can rely upon in those broader indications. So it's important to understand, however, that when companies haven't taken that step to produce the additional testing, we're left, physicians are left, patients are left, largely in ignorance or left to sort of guess about whether the product does work for these additional uses. And the scientific literature that studied off-label use has found that it generally lacks a scientific basis, albeit although it's actually very common in American clinics. And in fact, in some cases, especially in oncology and psychiatry and pediatrics, it's actually the standard of care, by which I mean it's what physicians expect themselves to do, even while at the same time it doesn't have a very good scientific basis to support it. And so this is just one of the recent studies of this literature showing that in about 12% of the prescriptions in this data set of over 151,000 prescriptions were they off-label, and the vast majority of those lacked strong scientific evidence to support the indication. Other studies have suggested a much larger proportion of cases are off-label, perhaps as many as a third of the prescriptions are written off-label. But the point here is at least that it is substantial. And if you look at the this subcategory of cases where they do lack strong evidence, and here I've carried forward the color scheme to this next figure, the study, Iguale and colleagues found that in these cases where there was off-label uses without strong evidence and support, there were much more commonly adverse events, patients that were actually harmed by the off-label drug. And so this is why, again, it's much safer and a better value to use drugs on label when we can, because companies have produced that evidence of science, safety, and efficacy that really motivates the FDCA in the first place, which is why we started there. Now, pivoting in the last couple minutes, I'd just like to talk a little bit about the application to COVID-19. We saw one particular class of drugs, hydroxychloroquine, used off-label, even though it, in contrast to it's fairly, it has multiple approved indications for malaria. This is interestingly in the media, everyone seems to mention it's a malaria drug, but in fact, it's also approved for the treatment of lupus and rheumatoid arthritis, as well, interestingly, as for prophylaxis of malaria in advance of someone being ill. You recall President Trump claimed, I don't know if it was ever confirmed, that he was taking this drug prophylactically for COVID-19, which would be another off-label use, in addition to its frequent use for treatment of COVID-19. You may have seen in the news just in the last 24 hours, 2 of the studies on that question were retracted for data problems. But one of the challenges then, if you have an approved indication, that means there is substantial evidence that it's efficacious for that use. In the last few years, we've also had huge problems of drug shortages, right? Where it's difficult to even get enough product into the clinic or into the hospital. And so one of the challenges facing, this is a figure from the Lupus Foundation of America, who's been really concerned that their hydroxychloroquine that their patients are depending on to treat their lupus is actually being diverted to COVID-19, where it's not known to be safe and effective, but really is being used much more speculatively. So this is an interesting example of a rare case where states have actually stepped up through either guidance statements or binding rules or executive orders to primarily limit pharmacists from dispensing hydroxychloroquine in these off-label indications in order to protect it for lupus patients instead. Okay, so if you're interested in more about off-label use, I'd recommend a paper of my own from Nejm with Aaron Kesselheim last year, a longer paper with Victor Lorian about this intended uses principle. And finally, just a little less than a year ago, the Congressional Research Service released this great report I'm putting here on the screen for your reference. Okay, thank you. Please follow me on Twitter, Prophecy Robertson, or follow up if you have any other questions, and I'll be here for the Q&A as well. Now I'll turn over to Allison.
Thanks, Chris. And I'm going to try to figure out how to navigate screens. The worst part of doing webinars from home is having to be your own tech support. So let's see if I can pull up my slides. There, hopefully everyone can see that. So I'm Allison Bateman-House, as was already said, and Chris just gave us a really great overview of what happens when you're bringing a new drug to market for a particular indication, and then when it's used for a different indication off-label. And what I'm going to talk about now is access to non-approved drugs, investigational medical products. And again, we were going to talk about in this section, new indications, off-label prescribing, emergency use authorization, and I'm hitting the pre-approval access area with specifically dealing with expanded access and right to try. And so why is this a thing? Well, it's a thing because, technological support. Because there are people who cannot get access to medicines. They're not available on the market yet. They can't get access to them in clinical trials, and they have a serious unmet need. And this used to be called compassionate use. And I'm sure everyone has seen examples of this in the media or newspaper stories, social media campaigns, where people say, I have no other option and I can't afford to wait. And I know it may not work, and I understand it may be risky, but let me have that opportunity. And you can just see from this graphic here, and Chris showed a similar one a minute ago, that in the process of bringing a new drug to market, yes, there are people who get access to that novel drug through clinical trials. In fact, that is the primary route of pre-approval access is clinical trials. But those numbers are relatively small compared to the larger population out there that may be waiting. So, again, you can see here, in a number of years that it takes to bring a drug to market, it's a relatively small number of volunteers who are able to get into those clinical trials. So that is why we have people who say, I still want access to this unapproved product. Can I please get access outside of clinical trials? And that's when we start talking about non-trial pre-approval access. And that's where you see campaigns like this. These are cases in which individual patients or their supporters or family or friends are asking a company, may I get access? And, this is not always the case. Sometimes these requests are very routinized. They don't need any sort of, public airing, but these are the cases that normally come before us when we do hear about, pre-approval, non-trial pre-approval access or compassionate use. And I just wanted to flag, it's a sticky situation because when we are testing a new product, that's research. We hope that people will benefit from being involved in those trials, but there's no guarantee. And in fact, that's not even the primary impetus of the trial. The primary impetus of the trial is to learn about the novel drug or to learn how it compares to some other product. It's research. The primary impetus is to learn scientific data that will help us understand the safety and efficacy of that drug for a particular indication in a particular population. And yes, we hope patients benefit along the way, but it's not the primary purpose. Whereas in treatment, the primary purpose is to help patients. And again, we might be able to collect some data along the way. You might be able to do some sort of chart review of patient records and be able to suss out, this drug works better than this drug, or this treatment approach works better, but it's not the primary intention. The primary intention is looking at the patient in front of you and saying, here's what I can do to hopefully benefit you medically. And so non-trial pre-approval access sits right in the middle here, and that causes issues. It causes issues just in terms of which legal structure, does it adhere more to in terms of institutional policies, which should we adhere closer to? And the issue that really concerns me as an ethicist is, how do patients understand what's happening? Do they understand that, you are being offered this product because we think it might have a treatment use for you? Are we offering you this product because it might have a treatment use for you, but really we're testing this to find out? Or are we sort of in the middle? So I will move on and just get to the actual regulatory definition in the United States. So non-trial pre-approval access is not in the Federal Code of Regulations. It's something we came up with to take the idea of compassionate use and come up with something that was a little bit more clear about what it's talking about. I could ask for compassionate use to an approved drug and say, I cannot afford this drug, on the grounds of compassion, won't you let me have it? So we were trying to be very precise about what we're talking about, which is non-trial pre-approval access. And it's access on the basis of medical need to an investigational, remember, unapproved medical product outside of a trial and before it is approved by a regulator. And so within the United States, the regulatory pathways that exist, there are two of them. The long-standing one that we've had in various forms since the 70s is expanded access, which is under FDA oversight. And the newer one, which I think just had its second birthday, is right to try as a federal law. And there are 41 state laws that are variants of that right to try law. And I'll just jump ahead and say, since, we are trying to tie this to COVID, right to try, as far as I know, is not being used in any COVID-related situation. So I'm not really going to spend too much time talking about it. Now, I just wanted to point out, I'm talking to you about the regulatory framework in the United States, but non-trial pre-approval access happens in many countries, not every country, but in many countries around the world. And there's myriad terminologies that are thrown about, which makes it really difficult for companies that are operating globally, for multinational patient groups, and just for people to be able to communicate amongst themselves. There's a plethora of terms, and it makes it hard to navigate. So some of them are on the screen, and certainly that's not a comprehensive list. But one thing that is in common between these various systems around the world is that non-trial pre-approval access normally has some similarities. And this is an article from Bunnick et al. In Health Policy. And these are the commonalities. It's limited to patients who are suffering from serious and or life-threatening diseases. So, this can't be, just a trivial matter. Standard approved treatment options must have been exhausted. So those drugs that Chris was talking about that have been approved for a particular indication, if that indication is your disease, you need to have tried those first before you can ask for non-trial pre-approval access to an unapproved product. There must be a reasonable benefit risk ratio. We think the possibility of it helping this patient is, greater than the possibility of it causing harm to this patient. Expanded access or any whatever one of these programs you want to call it, the non-trial pre-approval access program must be structured in such a way to not interfere with getting patients into those clinical trials, because clinical trials are where we get the data that pushes the field forward, helps us bring new drugs to market, and helps us be able to treat populations of people. And if you pull people off and say, why participate in the clinical trial when you can get access to you want outside of a clinical trial? It's going to have, systemic ramifications that are impact many other people. And finally, approval from a regulatory body is required. So those are sort of commonalities across most of these programs. Now, back to the United States again with expanded access. Some people don't understand that there are 3 types of expanded access. So when I showed those pictures to you a minute ago of people appealing to the public or appealing to a company and saying, please give me access to this product because it's my best chance, that's the individual expanded access, which you can see here on the slide. It's one person, and we're trying to help that one person. But companies have the discretion to actually set up a program and say, for a particular population of people that we think this is appropriate for, we're going to set this expanded access program up. Those can be intermediates, which I tend to very helpfully describe as somewhere between 2 to many patients, or treatment programs, which is, like many plus. You can actually have thousands of patients in a treatment expanded access program. And those are less known to the general public, but they also make up about 50% of the requests is that true? As soon as I said that, I'm questioning myself. Let me come back during the Q&A and make sure that that's right. But what I was trying to say is, this is not an atypical thing. It's quite common that there are these cohort expanded access programs as opposed to just the individual ones. And this all was very visibly played out, not only the existence of these various types of programs, but the confusion about it in the coronavirus context when Gilead's remdesivir suddenly became a very hot commodity. Because remdesivir was available both through clinical trials, which again is the preferred method of getting access to an unapproved product, and through individual patient expanded access. And what happened is Gilead was flooded with requests, each individual patient's doctor coming and saying, I have a patient, can I get access? I have a patient, can I get access? I have a patient, can I get access? And finally, at some point, Gilead said, look, we can't do this. It's not sustainable. We're putting a brake on accepting any more individual expanded access request with the proviso of if it's a pregnant woman or if it's a child under 18, we will still take those. But otherwise, we're not taking them. And what we're going to do is we're going to move to one of these cohort programs. And as soon as we do that, then we will reopen the program. And this was very not well understood in the media. And personally, I fielded many calls from people who were frantic about the fact that expanded access or compassionate use or whatever you wanted to call it to remdesivir was not going to be possible. And it was possible. It is still possible. So just very quickly, this is the process for a single patient request for those who are not familiar with it. The doctor and the patient together decide this is what they want to do. The doctor communicates to the company and says, I have this patient. This is why they can't go into your clinical trial. This is why I think it's appropriate. What do you think? The company can say no. And if they say no, that's where you get these sort of images I showed you earlier of patients appealing to, the public square to pressure the company. But if the company says yes, then the request moves to the FDA for that sort of risk-benefit analysis. And then finally, there has to be permission from the institutional review board at the patient's institution. Again, this can be for one patient or multiple patients. It can be for a product that's in any phase of development. The company can actually charge to reimburse its, to get its cost covered, its expenses of making this product available to the patient, but it cannot charge a profit. We can't profit off of unapproved drugs. Informed consent is required, and any serious adverse events that occur in this patient once they get the product have to be reported promptly to the FDA. Now, there was a campaign, as I said, it turned into federal law about 2 years ago. And prior to that, it led to a number of state laws called the Right to Try. And you can see that this campaign said, it's really the federal government that's keeping Americans from getting access to these investigational medical products that they might want through expanded access. So if you want to increase patient access, you have to get the federal government out of the way. And I just want to say, and hopefully you saw this a second ago when I went through my schematic, the gatekeeper is really the company. Companies cannot be forced to give access to their unapproved products away. They can be, pleaded with. They can have public pressure pressuring them, but legally there is no requirement to force companies to give access. So even though we have a law called the Right to Try, it's really a right to request access. And as such, the idea that all you have to do is get the federal government out of the way and you'll be able to get access was actually fundamentally misguided. And you can see that in these numbers that are reported by the Food and Drug Administration. You can see the vast majority of requests that come in, both for single patients and for these sort of cohort programs, they are, most of the time are allowed to proceed forward. And that's really because, it's the company that is the decider, and the FDA doesn't get these requests until after the company has already said, yes, I agree. So just to say for Right to Try, what are the differences? It's not for multiple patients, it's just for one. It's not for all phases of development. It's only after phase 1, which is known as sort of safety testing. Again, the company can only charge to cover its expenses. Again, informed consent is required, although what exactly that means is sort of a diminished standard from an expanded access. And serious adverse events must be reported, but only annually to the FDA as opposed to in real time. And as you can see, whereas you still have to have the conversation between the doctor and patient, and you still have to have the willing company, the FDA is removed as an oversight body. And I have sort of a dotted line over the IRBs because whether the IRB is involved or not really depends on sort of a hodgepodge of state law, company policy, et cetera. So much less, systematic approach than we see in expanded access. And so, what this all comes down to, my final 2 points I want to make about COVID is if you look at just this smattering of text that I pulled out from the last couple of months, we see in news articles, mention of Right to Try, compassionate use, single patient expanded access. And if you actually look into the stories, these are frequently used inappropriately. So I mentioned a minute ago, we don't see Right to Try being used in COVID-19. We do see single patient expanded access, but, perhaps in a certain circumstance, what is actually being referred to is a cohort expanded access program. Or in many circumstances, we hear about drugs being given compassionate use status when that's not what we're talking about at all. We're not talking about non-trial pre-approval access. These are drugs that are either off-label that have been given the green light to be used for COVID, or they're drugs that have been given sort of a provisional approval by the FDA, which Holly will talk about in a second. And then the last point I want to make is, you see these terms used again when we're looking at the actual attempts to collect data from the uses of these drugs. We are desperate to know what works, what doesn't work for what cohort of patients, when initiated when. And so any handing over of the drug, whether it's in a trial, whether it's non-trial pre-approval access, whether it's off-label, people have been trying to collect data from, but people who are then reading this, or the general public who are hearing this, frequently don't understand, the ramifications for, the level of evidence. Was there randomization? How trustworthy is this data? And we're going to have some people talking about that in just a minute. So I'll conclude there, and thank you for your attention.
Hi, everyone. I'm Holly Fernandez-Lynch, and I'm just getting my slides up. Okay, sorry about that. So, right, as Allison mentioned, I'm going to be spending a brief period of time, because I know we're running behind schedule, talking about a particular pretty infrequently used pathway called emergency use authorization. And just to kind of contextualize where things stand, Chris talked about off-label prescribing, Allison talked about pre-approval access outside of clinical trials. Emergency use authorization actually is a type of pre-approval access, and you'll see why momentarily. It is a pathway that is not the same as traditional marketing approval. And you'll see that I'm going to be quite careful only to talk about the emergency use authorization rather than approval, to differentiate between these two pathways. But what it does is allow for the use of products that have not been approved by FDA for any use, or for unapproved uses of approved products to be used widely by the population in certain types of public health emergencies. And the things to notice is that it's just a temporary authorization, and it can be used for a wide variety of what's called medical countermeasures in response to the source of the public health emergency. So you can have an EUA for drug, biologic devices. It can be for therapeutics, prophylactics, diagnostics, and I'll go through a couple of examples in a moment. So how do you get one of these emergency use authorizations? This pathway is actually not available in most cases. So, if I have metastatic cancer, certainly that's an emergency to me. It's an emergency to, my loved ones, but the emergency use authorization is not available for that particular indication. It has to be a particular type of emergency stemming from what's called CBRN agents. So that refers to chemical, biological, radiological, or nuclear agents. In the context of the novel coronavirus, right, that's a biological agent that's created the public health emergency that we're currently facing. But you could imagine, these other types of agents might be used for terrorism, and that's another function of the emergency use authorization. So you have to have the right type of emergency, and then members of the government have to make a variety of declarations in order to give FDA the ability to issue an emergency use authorization for a particular product. So first, you need to have a declaration of emergency, and that can be a current emergency, or it could be an anticipated emergency, so that you use the EUA kind of to prepare for future pandemics or future concerns of terrorism, for example. Who can make that declaration of emergency? Well, it can come from the Department of Health and Human Services. They have the authority to declare a public health emergency, but other agencies can also declare different types of emergencies. Homeland Security can declare a domestic emergency. Department of Defense can declare a military emergency. Once a relevant emergency has been declared, then the Secretary of Health and Human Services has to issue an emergency use authorization declaration, right? And that basically says to FDA, okay, you are now free to use this particular pathway to make products available. In the context of COVID-19, we had that HHS declaration of a public health emergency on February 4th. And then we had a couple of different EUA declarations for different types of products. So the most recent one was to allow FDA to authorize drugs through this pathway. In order for FDA to grant an emergency use authorization for a particular product, these 4 criteria need to be satisfied. First, the agent in question needs to be capable of causing a serious or life-threatening disease or condition. We know that's true of the SARS-CoV-2 virus. And then it has to be the case that it's reasonable to believe that that the medical product in question may be effective, either for prevention, diagnosis, or treatment of that disease or condition, based on the totality of available scientific evidence. And what I want to flag here is this standard of may be effective is different from the standard that's required for traditional marketing approval. That standard for marketing approval is substantial evidence of safety and effectiveness, typically from adequate and well-controlled trials. This standard may be effective is much lower, and it provides FDA with quite a bit of discretion to decide what evidence they're going to determine to be relevant. In addition to the product having to be possibly effective, it has to be the case that the known and potential benefits for that use of that product outweigh the known and potential risks, and there have to be no available alternatives. And I just want to highlight that there's a fair bit of overlap between these criteria and the criteria that Allison was describing for expanded access, especially when we start talking about expanded access for widespread treatment populations. Okay, so think, about the overlap: serious or life-threatening disease or condition, a risk-benefit analysis, absence of reasonable alternatives. The main difference for the EUA is that the intention is that you're a bit more confident about effectiveness. For single-patient expanded access, there's actually no requirement to demonstrate any level of effectiveness. And in the EUA, there's no automatic protection for clinical trials, right? Remember, Allison was saying for expanded access, one of the criteria is that you cannot participate in an ongoing trial. That's not necessarily the case for an EUA, although FDA can include that condition when it grants one of these authorizations. This is, the EUAs are intended to be distinct from research, so there is no IRB oversight, and there's also no limit on how much a company can charge for a product that has been granted emergency use authorization. And then I'll just mention, once those criteria are satisfied, FDA will put some parameters around the emergency use authorization, talking about which patients can use the product under this pathway, giving some information both for healthcare providers and recipients to promote informed consent and safest possible use, provisions for adverse event reporting, and then there might be some other constraints around how the product can be distributed, administered, and advertised. The EUA will be terminated automatically when the declaration of emergency goes away, right? So once we're no longer living in a state of emergency, anything that was still under an EUA is no longer approved or authorized for use under this pathway. A product that has an EUA, if you want to continue to be able to market it after the emergency is over, the sponsor will need to go ahead and provide the evidence and application necessary for traditional marketing approval. FDA also has authority to revoke an EUA if there's safety concerns. So this pathway is only about 15 years old. It's been used relatively infrequently. These are the diseases and conditions or kind of viral agents that have played a role in emergency use authorizations previously. But for the most part, the pathway has been used for diagnostic and antibody tests, as well as certain devices, including personal protective equipment. And very infrequently has it been used for therapeutics and prophylactics. Only once for a vaccine, and that was in the context of anthrax. And that was a vaccine that actually had been around for quite a while. It wasn't technically FDA approved for some historical reasons, but FDA has been pretty careful about using this pathway for therapeutics and prophylactics. That breakdown of use of this pathway is replicated in COVID specifically. So we had lots of EUAs and still have lots of EUAs for diagnostic and antibody tests. These were relatively controversial because at first FDA was, I think in many people's minds, too stringent about the tests that they were willing to allow to be used. And then they pulled back wildly and basically made it a free-for-all, which has caused problems for the validity and reliability of the tests that we have available. The EUAs have been granted for COVID in the context of ventilators, sterilization devices for PPE, and other devices. And we have 2 EUAs for therapeutic products at this point. Let me go through those in a little bit more detail. So the first emergency use authorization for a drug for COVID was hydroxychloroquine and chloroquine. And as Chris mentioned, these products were already approved in some formulations and from some manufacturers, but there was interest in using them off-label for COVID. And so you might ask, well, why was an EUA needed in this context if under the practice of medicine, physicians were able to prescribe these products? Well, the reason an EUA was needed was because it was limited to particular drug products, particular hydroxychloroquine and chloroquine from particular suppliers who had made their product available to the strategic national stockpile. And not all of those sponsors had FDA approval for that particular compound. It's also the case that when drugs are distributed from the strategic national stockpile, that's not practice of medicine, right? So the stockpile is going to start distributing these products to public health authorities around the country for an off-label use. And that would technically be a violation of the Food, Drug, and Cosmetic Act if it wasn't under this EUA. Okay, so it's just important to acknowledge that because the EUA was confined to only certain types of product distributed in certain types of ways, not all hydroxychloroquine and chloroquine was under an EUA, and you could actually get it outside of this particular authorization. This was granted on a very low standard of evidence. Remember the may be effective standard. Here, FDA was relying on limited in vitro and anecdotal clinical data, and it limited the use to certain types of hospitalized patients who couldn't participate in a trial. No process for collecting outcomes data as part of this EUA, and very basic standards for reporting safety concerns. So you may have heard that there's been a lot of consternation that the EUA in this context might have been granted as a result of political pressure on FDA. This is part of the former head of BARDA, Rick Bright's complaint. His whistleblower complaint was that they were pressured to proceed with this emergency use authorization. The evidence was very weak. And the fact that this was authorized in this fashion, even though it wasn't quite approval, was taken to, it looked like approval to many people. And so it looked like an endorsement of this product from FDA, despite the fact that we really didn't have evidence of effectiveness and there are some safety concerns. The next example of a drug that's been granted an EUA for COVID is remdesivir that Allison was talking about, first made available through expanded access, not yet approved for any use, but the EUA was granted in the context of a well-designed, randomized, double-blind, placebo-controlled trial that determined this drug can shorten recovery time from 15 to 11 days. So within a couple of days of those top-line results coming out from that trial, FDA was able to issue the EUA for this product, limited to, again, hospitalized patients in this context with severe COVID. There was no restriction in this EUA for trial participation. Regardless of whether you could be in a trial, you would be eligible for remdesivir under this EUA. I do just want to flag that there were distribution issues and continue to be distribution issues, right? So just because FDA says that a product may be used does not necessarily mean it's going to be available for use. Gilead Sciences has donated hundreds of thousands of vials of this product in the United States. At first, the federal government was handling distribution to healthcare facilities. That went remarkably poorly, and they determined that they would pass off that authority to local public health authorities. Lots of open questions about this product. Continued study is needed, but it was determined that there was enough evidence of benefit that it was appropriate to provide this through EUA. So just in terms of concluding thoughts, I do think this pathway can be a useful option to prepare and respond to pandemic circumstances, but it opens things up very wide to acceptance of weak evidence, and it can be a little bit hard to put the genie back in the bottle. It's important not to conflate EUAs with traditional marketing approval. The last thing I'll flag is, I mentioned it's only been used once for a vaccine. We know there's tremendous pressure, as Art described, to get a safe and effective vaccine to the public as quickly as possible. And so some people, including myself, are quite worried that this pathway will be used to authorize a vaccine. And if it's authorized under very weak standards, then all of those concerns that Art raised previously about trust in vaccines, false sense of security, those things will all come to light. And I'll close just with a couple of resources that you may find helpful. FDA has a guidance document all about emergency use authorization, and these other two websites are lists of the current emergency use authorizations as well as archived EUAs. So thank you very much, and I'll turn things back over to Kelly.
Okay, great. Thank you so much, Holly. So we're a bit over time, so we're going to save our questions until the end. I see that we have a lot coming in, and I'm continuing to collect them. So we will try to get to as many as we can at the end. So I'm going to go ahead and move on to our final panel and introduce them. So that topic is real-world evidence in COVID-19 treatment development, ethics, challenges, and considerations. We'll hear from some CUPA members who were also part of CUPA's sub-project on ethics and real-world evidence called IRWI. Dr. Barbara Redman, who we'll hear from first, is an associate of the Division of Medical Ethics at the NYU Grossman School of Medicine. Next, we'll hear from Dr. Haley Belli, an assistant professor in the Division of Biostatistics at NYU. And finally, we'll hear from Mr. Tom Watson, the executive vice president for early access programs at Bionicle EMIS, a global leader in running expanded access programs for pharma and biotech companies. Belli and Watson, along with Allison Bateman-House, are the co-chairs of our ethics and real-world evidence subgroup. Now over to Barbara.
Okay, let's go. Policymakers have long pushed for real-world data and evidence because of a couple of reasons of problems with randomized controlled trials. They had a lack of generalizability, they were expensive, and there was a long development time. So there had been a political push to move with use of real-world evidence and data for some while, which ended in the 21st Century Cures Act, when put it really on the front burner. FDA now accepts real-world evidence as supplementary evidence for product approval, and real-world evidence especially is used in post-marketing trials. So methodological work has been ongoing for some while, particularly in oncology, to work on methods for aggregating electronic medical records, payer data, assembling, curating all these data, providing common definitions, tackling inability to access some timely data, such as death certificates, or getting into the medical record, the fact that death has occurred. This movement has been heavy on methods and not very heavy on ethics. Ethical problems are still not well addressed. For example, there is a lack of representativeness of the data sources that I just described, which is payer data, electronic medical record data, because it's only certain patients who are insured that will be in those data sources. So far, there hasn't been much interest in moving to areas where patients are not very well insured, such as community clinics or other sources. So there's a real concern that real-world evidence, as it has been developed methodologically, is excluding a number of people. Also, you don't commonly find in medical records anything about social situations, which are terribly important. Now, enter COVID-19, and real-world data had to be used in parallel with clinical trials, especially in compassionate use. There was a sense that there was a moral obligation to collect data from all patients who had been treated with investigational products. And in clinical trials where randomization is unethical, in which there are small groups, or in personalized medicine, there has been a movement toward real-world data, real-world evidence. And FDA is moving with an initiative to capture real-world data and evidence for COVID-19. But in the midst of all this, there is no guidance on collection and analysis of the data in this situation with real-world evidence. But they're important. These data are important for risk-benefit decisions and for informed consent, both of which are strong ethical issues. So our panel today will be focusing on two issues that have to do with making meaning of the data. Haley will speak about statistical techniques to use on these data and how these techniques are underdeveloped. Tom will be talking about access and allocation of investigational products. And over to you, Haley.
Thank you for that introduction, Barbara. As a biostatistician, I'm now going to discuss statistical considerations when extracting real-world evidence from real-world data. And to begin this conversation, we need to talk about generalizability. So that is how representative a study sample is of the general population. So generalizability can often be thought of into two groups. So internal validity, which is the truth within a study, versus external validity, which is our truth in the real world. So if we're going to draw conclusions about real world evidence from real world data, we need to first assess the internal validity of the real world data or its source, and then we can look at the external validity of that real world data. Now, there are many factors that influence internal and external validity. For the purposes of today, I've decided to group them as follows, but in different contexts, they could, of course, be categorized as either internal or external or both. So first, one factor that may influence internal validity is how the population was recruited. So for example, were the patients volunteers? Was the population perhaps inpatient hospitalizations versus outpatient care, as that may indicate how healthy or less healthy the group may be? Or was the population truly recruited at random, in which case that would decrease bias the most? We also have to ask ourselves, what is the purpose of the data collection? So here we ask, what are the primary and secondary study outcomes, and do those align with the inference being sought? So a classic example is EHR data, which is often collected for billing and insurance purposes rather than research. So the data that you're looking at in the EHR may not necessarily reflect your primary and secondary research outcomes. We also need to consider what is the quality and type of data being collected. So in terms of an experiment, we might look at whether it's open label, single or double blind. Perhaps is the outcome patient self-reported, as that will introduce variability in how patients interpret, say, a questionnaire. And then also, as I just mentioned with electronic health records, because the purpose of collection might not be for research, the scope of the data that you're looking at may be limited or not even comprehensive. And then even if we're using sources such as tests or devices that do not have self-reported outcomes, every test has some type of error associated with it. So for example, as we're seeing with COVID, with the antibody diagnostic testing, we're not seeing 100% sensitivity. So no test is foolproof. And so we also need to consider that when looking at our results and interpreting them as evidence. And then finally, wrapping up for internal validity for now, patient follow-up is important. So we can't just look at one little snapshot in time of the data. We need to consider both long-term effectiveness and safety and adverse events over follow-up. So an example of long-term effectiveness may be with a vaccine. So this question of how long will immunity be provided? So maybe initially it provides some immunity, but is that still there 6 months down the road, 1 year from then onward? In terms of safety, we saw with research with the drug hydroxychloroquine, at first it was believed to show promise for efficacy, but then we started noticing these elevated risks for cardiac-related side effects. So only with following events up over time and seeing how patients are doing later, do we really capture this whole snapshot of evidence. So now switching our attention to external validity, one factor that we may look at is heterogeneity within the population. So differences across gender, socioeconomic status, the location, is it in a rural, suburban, or urban setting? We're noticing with COVID especially differences across all three of these variables. So it's important to consider which of those populations our outcome is actually in. Also looking at inclusion and exclusion criteria. So this is important because it tells us how broad or narrowly defined our study is. So for example, if the criteria is too restrictive, will have a patient population that may be healthy versus one that, if the criteria is broader, may represent a population with more comorbidities as perhaps more realistic or pragmatic. So that too may influence the study outcome and the evidence we're extracting. We also care about what the actual study design is. So often when looking at real world data, at best it's an observational study, usually just little snippets of data. But I've listed here also experimental clinical trial, because I think when we're considering ethical issues in COVID, it's important to also break down different phases of a clinical trial and what evidence they provide us with. So an example here is with the Moderna mRNA vaccine, which finished its phase 1 trial results that came out at the end of May. And from that, while it shows promise and is creating hope, there was no pre-human animal testing beforehand. And we know from research by Andrew Lowe's group out of MIT, in a paper that he published in the journal Biostatistics in 2018, they looked at the probability of success among nearly 2,000 different pathways from phase 1 to phase 2 for infectious disease vaccines. And over the course of about 15 years, only about 1% advanced from phase 1 to phase 2. So just because there's efficacy in phase 1 does not guarantee efficacy in phase 2, 3, and beyond. As a statistician, I always look at sample size. Simply put, more patients equals increased statistical power. And for those who are non-statisticians listening in, statistical power can be thought of as the likelihood that a study will detect an effect when there is an effect to be detected. And then depending on what our sample size is, we then go into the types of analyses or the methods used. And again, as a statistician, I often look for adjustments for covariate imbalance, either at baseline or for confounders that may arise during the course of the study, especially for observational studies and real world data. And so an example of that, we could go back to heterogeneity in the population. So for example, I may adjust in a model or look for that differences in gender or say socioeconomic status. And then finally, we hear the term over and over again of reproducible research. And that is so important for real world data and extracting real world evidence from it. Because especially if we have all these small snippets of data, we need to figure out some way to perhaps either pool those results together into a meta-analysis or different methods to validate that data by cross-checking, did independent studies give a similar result? So reproducible research is so important here. And so in conclusion, these are all types of questions that one could ask when attempting to answer whether any real-world data provides real-world evidence across any type of study. However, now that we're in the middle of the COVID-19 pandemic, of course, we're not going to have answers to all these questions. And so an important consideration is what is actually pragmatic when collecting real-world data. And so that leads us to our last speaker, Tom Watson. So take it away, Tom.
Thank you, Barbara. Thank you, Hayley, for your contributions to this section. And thank you to everybody for continuing to remain with us for this length of time. So this is the last session in this webinar, and it's only going to be a short presentation. For the next 10 minutes or so, I'm going to talk about real-world data with a particular focus on real-world data in the context of pre-approval access, and even more so in the context of COVID-19. So my name is Tom Watson. I have been a Cooper member for 5 years at the NYU, and alongside Hayley, I co-chair the Ethics and Real World Evidence Working Group. And I also work for a company called Bionicle EMAS, and we run global expanded access, pre-approval access programs on behalf of pharma and biotech companies. And indeed, 5 projects we have ongoing at the moment are related to assets and treatments for COVID-19, different treatments. We also receive thousands of pre-approval access requests every year, and across all of our programs, in one form or another, we are collecting data with a view that we turn that data into evidence that should have some utility. Before we move into real-world data in the context of pre-approval access, I felt that it was important, particularly given that this is the first in a series of webinars, to frame the discussion around some guiding principles. So a number of organisations, including the WHO, have published and laid out guidance as to some standards for research, access and supply in the face of a public health emergency like we're facing right now. These standards and ethical principles may be useful for yourself as either an individual or indeed you as an organisation to define what you see as important and the principles you'd like to adhere to, which is a healthy position to start from to make good decisions in the face of this pandemic. So the WHO has 9 ethical standards, which you can see on the screen. And whilst we don't have time to go through all 9 of those ethical standards, I would like to draw out some of the themes within. The standards do talk about international collaborative partnerships as it relates to companies and organisations working together. And that's something that I've been really encouraged to see throughout this pandemic, more than I've ever seen actually, as it relates to the development of treatments and vaccines. And hopefully that bears well for us to get some treatments sooner rather than later. And I know the first successful treatments are obviously starting to emerge. Data collection, and particularly real-world data collection and clinical trial methodologies in a period like we're in right now can be and should be pragmatic. And Hayley talked a lot about this topic in her previous discussion. But at the same time, it's really, really important that any data that we glean for the benefit of future patients is really meaningful. So a lot of thought needs to be had in relation to what that methodology looks like. And whilst informed consent for real-world data collection in general is really, really challenging, if you layer onto that COVID-19 complications, then it's extremely difficult. That said, it remains a fundamental ethical requirement for us as it relates to clinical studies and data collection. So if you do have a chance, I would encourage you to review those 9 ethical standards laid out by the WHO. So another really useful tool that may prove useful for your decision making during the COVID-19 pandemic was published by Johnson & Johnson, and it relates to supply, relates to access, and also research in the area. And they broke down some priorities as it relates to supply of medicines into priority order, which I, 1, agree with, and 2, find really, really useful. The first priority is to maintain supply for patients who are currently on approved treatments, which makes a lot of sense for a number of different reasons. And continuity of supply has to be an overriding principle. The next priority in relation to supply is to try to provide access to patients that are in randomized controlled trials. And again, that makes a huge amount of sense so that we can start to establish whether drugs are safe and effective. And that then generally is the way that drugs are approved, and indeed the way that broader groups of patients get access to treatments in the long run. So randomized controlled trials have to be a priority for us when we think about supply, particularly when we have limited stock available in many situations in COVID-19. And then the third priority relates to, I guess, research and also registries, etc., and studies that are non-randomized clinical trials. So clearly a lower priority, but it is the third group that I'd like to focus on for the remainder of my slides. because I spend my time working with companies running pre-approval access programs, and I believe that data from those programs, if designed well and planned for well, can be really meaningful, as long as we're adhering to the principles that I've already established. You would have heard from Alison earlier as it relates to some of the principles surrounding pre-approval access. And just as a reminder of some of those principles, the primary objective for this type of access is treatment and not research. Secondly to that, it can in no way hinder patients getting onto clinical trials. And clinical trials, as I've mentioned previously, should be the priority over pre-approval access programs in terms of patients entering them. And then thirdly, in no way can a pre-approval access program hinder development of a program. All that being said, I believe that if planned for early enough and if really well designed, a pre-approval access program can be a really good source of meaningful data that can complement and supplement randomized clinical trial data and evidence. And indeed, if designed well, pre-approval access programs can in fact direct patients towards clinical trials rather than jeopardize clinical development in any way. So now just moving on to some practical considerations as it relates to real-world data collection in pre-approval access programs. First and foremost, really careful consideration needs to go into what we're trying to achieve, how the data from the program will complement the clinical development program. Are we trying to collect data and turn that into evidence so for regulatory purposes or for reimbursement purposes, or to try to broaden the evidence for a particular group of patients? Clearly, given that access and treatment is the primary driver of pre-approval access, any kind of data collection needs to be balanced with the additional burden that is putting on healthcare professionals and patients. And generally we land in all of our programs around the collection of an additional 5 to 6 data points that might be meaningful and useful. However, in a COVID-19 situation and in some healthcare systems, you know, in ICUs with horrendous conditions, that might even be a stretch too far. And certainly we do not want to be placing additional pressures and burden on healthcare systems and professionals. So that needs to be really taken into account. Each country has varying different regulations about what can be collected from a real world data point of view, and these need to be taken into account. As a good example of that, Italy has very clear guidance that in order to collect data from a pre-approval access program, you need to go through a specific regulatory route, the compassionate use protocol route, rather than other routes available. So they need to be taken into account, and they will differ country by country and depending on the situation also. I think over the course of the pandemic, the largest criticism that I've seen related to compassionate use in particular data has been the absence of or the lack of a suitable control group. So a huge amount of planning and thinking needs to go into what a suitable control group might be. Now, it's not going to be perfect because these data have severe limitations, which I've tried to stress today. But nonetheless, if we are going to collect data and then try to get some meaningful outcomes for it, we do need to think carefully about control groups that we're using. And then we can't ignore, just because we're in a state of emergency, very, very important topics like consent, which I've talked about previously, and of course, data privacy, which is more important than ever, you could argue. And that leads me then on to the topic of what to collect. And unfortunately, the answer is not straightforward here. It really, really does depend on firstly, what you're trying to achieve, collecting data from any kind of program and what that data will be used for. So that needs to be planned for really well. But it also depends on the clinical setting. What you would collect in ICU is obviously very different to what you would collect in a community setting. And standard of care across countries and across health systems can be extremely different with different infrastructures, meaning that your collection strategies and real world data strategies need to be totally different as well. And we've already talked about the need for sensible and appropriate synthetic control groups. So whilst that was very short, hopefully it gave a snapshot of some considerations as it relates to real-world data collection in the context of pre-approval access programs and in the context of COVID-19. And I look forward to getting into more detail at future series of these webinars. That concludes the first episode of this webinar series. And we really hope that you've enjoyed the time and all the presentations today. At this time, we would really like to invite you to submit questions. And our moderator, Kelly, who's wonderful, is going to lead the Q&A session. So thank you very much for your time.
Thank you so much, Tom, for a wonderful overview. So we're going to spend the next 10 minutes with all of our panelists answering some of your questions, and I'll do the best I can to get to as many of them as we can. We just had this very interesting and timely question come in, and I'd like to direct this to Chris Robertson first. What would be effective recommendations to collect data on uninsured populations, especially populations where COVID-19 morbidity is significantly higher? I ask because as the weeks have passed, there's a lot of data suggesting the African American community is being hit harder than, say, the Caucasian population. And given recent events in the US, it is important to address this.
Yeah, this is a real challenge to collect data through the healthcare system for populations that don't have access to the healthcare system. So it is a bit of a chicken or egg sort of challenge. You know, there is this parallel in parallel to healthcare. We have a public health infrastructure in the United States, organized at the county and state level, and to some degree the federal level. And so there have been, you know, longstanding efforts at public health surveillance that go alongside healthcare. And so this is an opportunity to invest in, I think, public health surveillance, while also trying to continue to open up our healthcare systems through qualified community healthcare centers and other sort of public health access points. But I think the challenge is collecting data rigorously outside of clinical trials and outside these networks. The Veterans Administration has been another resource for that particular subpopulation that a lot of valuable data has come out of in years past. So there's a few thoughts off the top of my head.
Great, thank you so much, Chris. Allison, I know that you also wanted to get in on this question.
Yeah, I just wanted to point out, I show video. I just wanted to point out that Dr. Aisha Langford at NYU and I have a paper, a blog post coming out next week, I believe, in Health Affairs, that talks about the necessity in a situation like COVID-19 where you do have disproportionate disease prevalence in, you know, the uninsured, prisons, large congregate populations, to really push energetically those clinical trials out of just your academic, medical centers and into, your federally funded healthcare centers, prison hospitals, and other, not common sites of research, just to make sure you do get that data in the populations that are most impacted. Thank you, Kelly.
Great. I see we have some new questions coming in. I do want to get to a few from earlier, as we've had a few themes. One of them is advice on allocating a limited supply of an agent in the context of a pandemic. I guess I'll address this to Art first. More specifically, if there are any international organizations that could assume the role of figuring out how to equitably and ethically distribute limited doses of a COVID-19 vaccine.
Thanks, Kelly. This is a very easy question. As of right now, no. WHO has no enforcement. We may not be, the USA, a member for much longer. Even in the US, I think the decision about distribution is likely to fall to governors. There is no CDC can advise, but it can't enforce. We had this situation when we had a shortage of flu vaccine way back in 2004 due to a manufacturing problem. And it was first comes, first serve, and people were running around. Even a black market appeared. So unless we set up either an international group or a national group, I think we're in for some rough times on distribution.
Thank you so much, Art. Turning to our real-world evidence panel, we have a question about that. When considering real-world evidence, what are you considering to be a control group? And how can you avoid important issues such as confounding in assessing efficacy? So I think I'll direct that question maybe to Haley first, and Tom, Barbara, if you'd like to chime in, that would be great.
Yeah, so in terms of having a control group, I think they use a lot of strategies that you sometimes also see in RCTs. So rather than having an active control group, you can call upon historical controls. You can look through the literature of observational studies and then use perhaps those controls that appear in those studies to serve as your controls. So there's kind of a whole body of literature of ways to mimic RCTs using various observational data available. So yeah.
And Kelly, just to add to that, it's Tom here. From my perspective, as I said in my talk, this is a not straightforward question. And I guess using a controlled group does not turn a real world evidence study into a randomized controlled clinical trial. But I think most of the criticism so far of, in particular, compassionate use programs that have tried to glean some evidence from them is that there was no thinking at all that's gone into a control group. So I think in the right context, these data can be valuable and meaningful, but they obviously do have severe limitations, as Haley's mentioned. That said, it doesn't mean to say that we give up on the data and we don't try to learn from every kind of access interaction for the benefit of future patients. So I think we need to recognise the limitations. And as I said, doesn't mean to say these real world evidence interactions turn into randomized controlled trials, but nonetheless that also doesn't mean to say that they're not meaningful in any way.
Thank you so much, Tom and Haley. We have another very timely question coming in. I think I'll maybe direct this to Holly, but others can chime in as well. What do the recent retractions of the Lancet article on hydroxychloroquine and the New England Journal of Medicine articles on ACE-ARB inhibitors, both of which were observational studies, say about the role of real-world data and evidence in COVID-19 and regulatory decision-making?
Thanks, Kelly. Yeah, I'll pass the observational part of that and the real world evidence part of that to the other folks who are presenting. The comment that I'll make on the retraction of these articles is just about the speed that we're seeing in allowing new data to be released through medical journals. And that's not even to talk about preprints, but it's becoming, I think, increasingly obvious that we just have an example of fraud that took place with regard to those two articles, and research misconduct on the part of the authors who signed their name to these articles without clearly ever having seen the raw data. So I don't know that it's something that would be unique to observational data or anything of that nature. I think it's more a comment on the speed at which everybody is trying to move in the pandemic. And as a side effect of that speed, we're actually delaying things further because we're adding confusion into the mix of all of this, where we get data, we don't know how trustworthy it is, we have to pull back on it. And that's a real problem in the context of a pandemic.
And this is Barbara, and I agree with Holly. And it's in addition, it's possible that the peer review system has lower standards or is not able to function with the overload. So those are all problems.
So I've got one more question that was submitted earlier, and I think that will be the last one we'll be able to get to. Considering the history and the context of the pandemic and the level of desperation there is to get new treatments available, I guess I'll open this to any panelist who wants to answer. Do you have concerns that the FDA will lower standards of approval for a vaccine or treatment that may increase risk beyond the typical conservative way that we might think of risk-benefit ratios outside of the context of COVID-19?
Yeah, this is Art. I'm worried that we could see sort of an October surprise on the release of vaccine. I saw just now as we've been meeting, Trump announced he had 2 million doses of vaccine ready to go. Now that's 2 million doses of unapproved, untested vaccine. Somebody may have made it, but it doesn't mean it works or it's safe. So I think it's partly FDA wanting to get a vaccine, for example, out there, but it's partly political pressure. And that is out there. Look, Trump wants to be reelected. He's got a pandemic on his hands. Now he has these, protests and charges of racism flying at him. He's got to get the subject changed. And ideologically, that's a time to bring big pressure on FDA to approve something in time for the election. So I am worried about that.
Thank you so much, Art. So it is 3 p.m. now. I want to thank all of our panelists for giving us their time today to provide their expert presentations on some of these very important issues. So episode 2 of our webinar series is going to be held on Friday, July 17th. The topic is still to be determined, but we will send everyone who participated in today's event information on the next episode when it's available. Thank you so much for joining us, and we hope to see you next time.